What we need to know first, what is primary productivity? Primary productivity is the synthesis of organic compounds from atmospheric or aqueous carbon dioxide. It principally occurs through the process of photosynthesis, which uses light as its source of energy, but it also occurs through chemosynthesis, which uses the oxidation or reduction of inorganic chemical compounds. Almost all life on Earth relies directly or indirectly on primary production. The organisms responsible for primary production are known as primary producers or autotrophs, which form the base of the food chain in both terrestrial and aquatic ecosystems. There are two types of autotrophs, photoautotrophs and chemoautotrophs. Photoautotrophs rely on light for energy and use photosynthesis to produce organic carbon compounds, and chemoautotrophs rely on energy from chemicals, mainly inorganic substances such as hydrogen sulfide and ammonium. The production of these relatively simple molecules may be used to further synthesize more complicated molecules, including proteins, complex carbohydrates, lipids, and nucleic acids, or be respired to perform work. Consumption of primary producers by heterotrophic organisms such as animals then transfers these organic molecules and the energy stored within them up the food web, fueling all of Earth's living systems. Primary productivity varies among ecosystems and depends on many factors. In a terrestrial ecosystem, these include solar energy input, temperature and moisture levels, carbon dioxide levels, nutrient availability, and communal interactions, e.g., grazing by herbivores. These factors affect how many photosynthesizers are present to capture light energy and how effectively they can perform their role. A single factor that reduces a significant proportion of primary productivity yield is photorespiration. For example, this can be as much as 20% in wheat and 36% in soybeans. Photorespiration reduces primary productivity. Photorespiration refers to the process in plant metabolism where the enzyme ribisco oxygenates ribisculose 1,5-biphosphate, causing some of the energy produced by photosynthesis to be wasted. The desired reaction is the addition of CO2 to RUBP, carboxylation, a key step in the Calvin cycle. However, approximately 25% of reactions by ribisco instead add oxygen to RUBP, oxygenation. The oxygenation reaction of ribisco is a wasteful process because 3-phosphoglycerate is created at a reduced rate and a higher metabolic cost compared with RUBP carboxylase. As you can see, in photorespiration, instead of fixing CO2, ribisco fixes oxygen with the RUBP and therefore doesn't gain carbons and is unable to produce 2 times 3 glycerate molecules and instead produces half the number of glycerate molecules and a phosphoglycolate, which is unable to be broken down for energy. While photorespiration results in the formation of glyceraldehyde triphosphate, eventually there is a net loss of carbon. This typically happens in hot conditions because ribisco has a high affinity to O2. Ribisco is also more likely to fix O2 in increased temperature because a plant will likely close its stomata to preserve water, and this reduces ability to take up CO2, and O2 is unable to diffuse out, therefore resulting in an increase in O2 to CO2 ratio. Some plants, unlike wheat and soybean, can escape the worst effects of photorespiration. The C4 and CAM pathways are two adaptations that allow certain species to minimize photorespiration. All plants ingest atmospheric carbon dioxide and convert it to sugars and starches through photosynthesis, and they do this in different manners, including C3, C4, and CAM pathways. The specific photosynthesis method or pathway used by the plant classes are variations of a set of chemical reactions called the Calvin cycle. C3, C4, CAM plants. C3 plants. C3 plants are lacking photosynthetic adaptations to reduce photorespiration. About 85% of the plant species on the planet are C3 plants, including rice, wheat, soybeans, and trees. C4 plants. In C4 plants, the light-dependent reactions and the Calvin cycle are physically separated, with the light-dependent reactions occurring in the mesophyll cells and the Calvin cycle occurring in special cells around the leaf veins. These cells are called bundle sheath cells. The C4 pathway is used in about 3% of all vascular plants, including sugarcane and corn. C4 plants are common in habitats that are hot, likely because in hot conditions the benefits of reduced photorespiration exceed the cost of ATP used to move CO2 from the mesophyll cell to the bundle sheath cell. First, atmospheric CO2 is fixed in the mesophyll cell to form a simple 4-carbon organic acid. This step is carried out by a non-Rubisco enzyme, PEP carboxylase, that has no tendency to bind to O2. This 4-carbon organic acid is then converted to a similar molecule, malate, that can be transported into the bundle sheath cells. Inside the bundle sheath, malate breaks down, releasing a molecule of CO2. The CO2 is then fixed by Rubisco and made into sugars via the Calvin cycle, exactly as in C3 photosynthesis. CAM plants. Some plants that are adapted to dry environments, such as cacti and pineapples, have the CAM pathway to minimize photorespiration. Instead of separating the light-dependent reactions, and therefore the use of CO2 in the Calvin cycle in space, CAM plants separate these processes in time. At night, CAM plants open their stomata, allowing CO2 to diffuse into the cells. This CO2 is fixed into oxalate by PEP carboxylase and then converted to malate. This organic acid is stored inside vacuoles until the next day. In the daylight, the CAM plants do not open their stomata but can still photosynthesize. This is because the organic acids are transported out of the vacuole and broken down to release CO2, which enters the Calvin cycle. This controlled release maintains a high concentration of CO2 around the Rubisco. Plants use the total energy fixed by the three pathways, C3, C4, and CAM photosynthesis, as maintenance energy. The remainder is used for new plant biomass and reproduction and is food for herbivores. The amount distributed and the rate of turnover of biomass determine many important characteristics of the community diversity of herbivores and activity of decomposers. These relationships are constrained by the efficiency which plants absorb light, incorporate absorbed energy into photosynthate, and convert photosynthate into biomass. Productivity is measured by several means. A. Determining the rate of photosynthesis and respiration. B. Estimating changes in biomass over time. C. Correlating biomass to an easily measured variable to estimate standing crop. D. Use of computer models to determine productivity over broad spatial scales and predict response to changing environmental conditions. Productivity in terrestrial ecosystems are influenced by numerous factors: carbon dioxide, light, temperature, moisture, nutrients, and soil texture. The more productive ecosystems are those with optimum levels of such factors, ultimately to maximize photosynthesis and thus primary productivity. These factors exert their influence simultaneously and interactively, often doing so in non-equilibrium manner. At lower latitudes, we can see that productivity increases as plant growth is much higher in these regions compared to higher latitudes.
What is gross primary production? Gross primary production is a carbon dioxide flux, the uptake of carbon dioxide in photosynthesis and its release in respiration. Gross primary production plays a central role in the global carbon budget, and thus there has been an increased need to understand its variability globally. The impact on climate on the terrestrial carbon cycle is a focus of global climate change studies, resulting in advances in the quantifying and understanding of spatio-temporal patterns of the terrestrial gross primary production with ground, atmospheric, and space observations. Plants exposed to elevated CO2 show increased rates of photosynthesis and plant growth, and lowered stomatal conductance. The effect of CO2 concentration on photosynthetic rate for C3 and C4 crops is shown. C3 plants typically respond better to atmospheric CO2 enrichment than outdo C4 plants in terms of increasing their rates of photosynthesis and biomass production. Hence, it has been suggested that in the world of rising atmospheric CO2 concentration, C3 plants may outcompete C4 plants and displace them, thereby decreasing the biodiversity of certain ecosystems. However, the story is much more complex than what is suggested by this simple scenario. A study by Goodfellow et al. Compared plants grown in CO2 enriched and ambient atmosphere conditions during long-term exposure in the wet tropics of northern Australia. This study shows that an increase in CO2 concentrations, we would expect to see an increase in overall biomass, which is a result of primary productivity. However, this would be affected by other limiting factors such as light, nutrient and water availability. Their study shows that an increase in CO2 will have a positive effect on root and tree dry weight. Over the past century, the Earth's atmosphere and biosphere have experienced much change. Since the dawn of the Industrial Revolution, the mean global CO2 concentration has only risen. The rise in atmospheric CO2 concentrations is occurring due to the imbalances between the rates that anthropogenic and natural sources emit CO2 and the rate that biospheric and oceanic sinks remove CO2 from the atmosphere. With past deforestation and current trends of losing biodiversity, productivity of systems around the world will continue to decline. Loss in plant species like trees, grasses, ferns, etc. in forested ecosystems will reduce individual plant productivity by affecting both resource acquisition and efficiency of resource utilisation. Primary productivity estimates are collected from two different observation-based data sets, Model Tree Assemble and Moderate Resolution Imaging Spectroradiometer, MODIS. The first one is a data-driven model based on eddy covariance data, and the second is a data-driven model based on satellite measurements. The Model Tree Assemble uses eddy covariance measurements from data collection flux towers, global satellite retrievals measuring the fraction of absorbed photosynthetically active radiation, and global climate fields to provide global gross primary productivity. Around the world, there are 600 tower stations measuring carbon dioxide exchange between ecosystems and atmosphere, from which an algorithm can be applied to achieve estimates of global gross primary productivity. Most of the eddy covariance stations available are in Europe and North America, so the gross primary productivity is poorly constrained and there are regions that are not covered by measurement stations. Furthermore, the contribution of C4 plants to the global gross primary productivity is not included in the eddy covariance model. And another source of uncertainty in this model is that carbon dioxide is not increasing in time, which we know it is. The second gross primary production product uses satellite measurements gathered using MODIS, which provides quantitative and dynamic measurement of spatial and temporal productivity by vegetation. Using satellite imagery shows reflectance of plants, therefore a reduction in plant growth caused by deforestation, desertification and disturbance like fires, insect outbreaks and storms, or the enhancement in vegetation growth caused by reforestation, fertilizer use and the effects of nitrogen deposition will be identified and used in the gross primary production estimate. Global spaceborne observations of solar-induced chlorophyll exhibit a strong linear correlation with gross primary production, as you can see in the figure. Examples of how satellite imagery is used to estimate gross primary production, we have shown Australia's vegetation carbon uptake. You can see how it fluctuates each year. We have also compared the gross primary production in 2016 for the Eyre Peninsula and the Adelaide Mount Lofty Ranges, and looked at what months the vegetation carbon uptake was the highest. The analysis has shown that the Eyre Peninsula has a lower gross primary production compared to the Adelaide Mount Lofty Ranges. The Eyre Peninsula had a vegetation carbon uptake of 360 grams of carbon per metre square, and the Adelaide Mount Lofty Ranges was 900 grams of carbon per metre square. The Eyre Peninsula vegetation carbon uptake peaked late August, where the Adelaide Mount Lofty Ranges peaked early October. To assist in why this might be the reason, we have shown the leaf area index and the proportion of different land cover types. that both contribute to the overall gross primary production estimate. As you can see, the leaf area index is much higher for the Adelaide Mount Lofty ranges, and the land cover types vary between the two regions.